Okay, so I am doing my presentation on the genome editing technology, in specific CRISPR. So what is genome editing? It is the technique used to modify the DNA in the cell. This process can change the characteristics of an organism. It involves making cuts at specific DNA sequences with enzymes called engineered nucleases. Genome editing can be used to add, remove, or alter the DNA in a genome. And for the innovative technology of CRISPR, CRISPR is the most common, cheap, and efficient system used for genome editing. We're going to start with precursor technologies beginning in the 1950s. Arthur Kornberg would be the first person to develop DNA in a test tube. He had been working on the project of DNA synthesis from about the early 1950s. When he discovered all 5 nucleotides could be synthesized in a lab, he decided to turn his focus to the remaining factors needed for DNA synthesis. And he isolated DNA polymers from bacterial extracts, and within a year successfully synthesized DNA in virtue for the first time. The second big discovery was DNA ligation links. So the discovery of DNA ligase is considered a pivotal point in molecular biology because they're essentially essential for the repair and replication of DNA in all organisms. And this catalyzed further discoveries. And then in 1970, we have the purification of type 2 restriction enzymes. So a team was able to identify 6 base pair phage DNA sequences. This understanding of how restriction enzymes cut DNA and how host DNA works to protect itself is the basic for modern genetic engineering therapies that are going to be developed, and this is crucial for the technology of CRISPR. The last one we have in 1972 was what everyone was waiting for, was the discovery of rDNA. So rDNA essentially is DNA that has been created through the combination of elements of DNA from different organisms. By introducing genetic material from one organism to another, this discovery established the principles of modern genetics and was the basis for many future improvements. On this side, that one is definitely going to be the most important and relevant in CRISPR technology. Moving on to the 1980s. The 1980s was a major breakthrough period in genetic engineering timeline. During this time, a lot of ideas were turned to real products and ideas and really set the tone for the next century. The first would be the transgenic animal in 1981. So this is any animal that has a gene from a foreign organism deliberately inserted in its genome. This process first began with a research team led by Thomas Wagner at Ohio University. Wagner and his team transferred the gene of a rabbit into a mouse genome by using a method now standard in genetics known as DNA microinjection. And this also paved the way for animal experiments in the years to follow. Second would be in 1982 with the first synthetic drug. This is also going to be a big breakthrough. A company in the biotech game for over 40 years, Genentech scientist Dennis Clyde helped put the world's first genetically engineered drug, insulin, on the market for humans. So insulin had been used to treat patients suffering from diabetes, but now it was synthetic and easier to use. In 1983, the development of the polymerase chain reaction comes into play. So this was integral into later DNA experiments and breakthroughs, but it's still important at the time. It is used to make copies of specific DNA segments by implementing a chain reaction of any copy of a DNA sequence. It's amplified to make more copies, so it's easier now to make copies of DNA and can generate thousands or even millions of copies, as said before. So this makes it more doable than it was before. Then we can look at the discovery of zinc finger nucleases in the 1980s. This was highly targeted at genome engineering. The discovery of zinc finger nucleotides improved the effectiveness of gene targeting in several ways. So it made it easier to recognize DNA, which was the biggest one. And then this new technique helped promote the ideas of backward genetics over forward genetics, which was also a big breakthrough for future uses of CRISPR. Moving into the 1990s and early 2000s, we have the discovery of the principles of CRISPR and its functions. So this was done by Francisco Mageco, who was the first researcher to characterize what is now called a CRISPR locus. This took place at the University of Alacantí in Spain. He worked on this project throughout the 1990s and 2000s, and he recognized that he had been reported as disparate repeat sequences actually shared a common set of features known as the hallmarks of CRISPR sequences. So this was a major breakthrough for CRISPR. In 2005, the discovery of Cas9 and PAM was made. So this was done by Alexandre Bulletin, a French researcher at the French National Institute of Agricultural Research, who was studying bacteria. When he just made this discovery, although the CRISPR array was similar to previous reported systems that had been discovered, it lacked some known Cas genes and contained, so essentially he discovered Cas and PAM, which was separate from the CRISPR that had been discovered before. Moving on to 2007, a major discovery. This was an adaptive immunity, so the application to drugs was by Felipe Hartro. Him and his colleagues showed experimentally that CRISPR systems are indeed an adaptive immune system. They integrate new phage DNA into the CRISPR array, which allows them to fight off the next wave of attacking phage. So this is done in dairy products, this experiment. And then it showed that the Cas9 is likely the only protein required for the interface, which also makes it a cheaper technology to be used. And then in 2008, we have CRISPR acts on DNA targets. And this is crucial because before it was the RNA. Slide 5, we have the technology in the future. So CRISPR-Cas9 harnessed for genome editing. And this major breakthrough was made by Fen Zhang at the Broad Institute of MIT and Harvard. So another university sponsored finding. And he was the first to successfully adopt CRISPR-Cas9 for genome editing in eukaryotic cells. Next, we're going to look at where this funding for this research comes from. Obviously, you can see in a lot of the discoveries that the universities of these scholars funded them. The first major discoveries were done in Europe. Then, as you can see, we move into large universities like MIT in the United States. So they funded their college's research on CRISPR-related projects. The next is the NIH, which had recently increasingly begun to fund CRISPR research. They have funded over 6,500 projects between 2011 and 2018, and they have given over $3.08 billion in this 7-year period to research of the many different research projects. This funding is in the hopes of finding new technologies that will treat diseases, improve crop production, and create new sources of energy. So this has nothing to do with human genomes. The third is CRISPR Therapeutics, and this is an independent public company, Swiss-American company founded in 2013 that has given over $127 million in research funding to CRISPR research. So now we're going to look at the potential groups of CRISPR. First, we're going to start with the innovators. From the timeline, we can see that university-funded research. So these are the people who discovered the CRISPR. The early adopters, as you can see, were the synthetic drug producers, in specific, using genome editing to create insulin. This wasn't exactly CRISPR, but these were the early adopters of this. Next, I'm going to look at the early majority, which is GMO foods. I'm sure everyone's heard of GMO foods before. This would be an early majority since it is now a widely used thing. This technology is aimed to make crops more productive, last longer shelf life. The late majority, we're now starting to see the allowance of animal models. This would be the later majority because of FDA regulations. And it is kind of a precursor for as it moves, this CRISPR technology moves into humans, which moves me to the next one, the laggards. Due to the restrictions made by the FDA, my prediction is that these will either be the laggards or may not happen at all. There's been much controversy over the ethics of applying CRISPR technology to the use of the human genome, just because it involves cloning and changing human DNA. A lot of my research found that we don't want to become like the people of Brave New World. Third, we're going to look at the conditions and effects on diffusion. So mainly this is just FDA regulations because this is working with genes. The biggest setback for the CRISPR technology is definitely the FDA. It must pass the FDA regulations to be diffused into the market. So obviously the industries with the most ethical issues would be using this technology on human genes. So it's going to take them the longest time to diffuse this technology into the marketplace. However, technologies that CRISPR is already being used for would be food and drug production, because these gene editing technologies are not that new in those divisions, as well as other sectors like fuel. And that is all I have.